So, good afternoon, everyone. We are letting people in as they come in. But thanks for joining. We're really excited that you're all here. We'll give folks another minute or so, and then we'll go ahead and dive in. Um, we will explain what we're doing here, what this webinar is all about, why you're all here. We'll talk about some housekeeping, then we'll introduce ourselves and our wonderful panelists, dive into some presentations. We will save questions for the end and we'll explain exactly how to ask those questions. Um, so as people still trickle in, Lacey, I think we can go ahead and proceed. Okay, so housekeeping, very important stuff. So we are recording this webinar. It will be made available in the sense that if you're interested in the recording, you can go ahead and ask for it. It will not at this point be posted anywhere publicly, but if you'd like the recording for yourself or for your organization, you can make the request afterwards. As I said, so this is a webinar style presentation, so we're only going to be taking questions via the Q&A feature, which you can find down at the bottom of your screen. It should look like that little Q&A icon, and you can enter in your questions there. So I'm sure as our panelists speak, there will be questions that come up. Go ahead and enter your questions in as they come up, but know that we won't actually be addressing them until the very end when we'll have the Q&A session. At the end, we'll ask you to complete a short survey. So once you click out of the webinar, you'll get a little screen that pops up that takes you to the survey. It will also come to you in the email that you registered with. Okay. Next. So welcome to State of the Science PFAS in Local Foods, Understanding Exposure from What We Grow, Hunt, and Harvest. This is the second webinar in a series of three. And once we introduce some of our personnel, our wonderful Community Engagement Director, Liz, will explain a little bit more about why we chose to have this series of webinars, why now, why these topics. But this webinar series is being hosted by Duke University Superfund Research Center, the North Carolina State Center for Human Health and the Environment, North Carolina Coastal Federation, and Cape Fear River Watch. Next. Okay, so who are we? So we have two centers who are going to be facilitating Duke Superfund Research and Center for Human Health and the Environment, designated by the dark blue and the lovely green, respectively. So we have Dr. Katie May, who is the co-director of Community Engagement Core for the Center of Human Health and the Environment, and myself, who is the program coordinator for community engagement at the Superfund Center. Joined by our wonderful colleagues, Lacey Brown, Community Engagement for the CHHE, Dr. Liz Shapiro Garza, who is our Community Engagement Director for the Superfund Center, and Megan Hort Hughes, who is our Senior Program Coordinator for Research Translation. Myself and Dr. May will be facilitating. So why this seminar series? And I'll pass it over to Dr. Liz Shapiro Garza.
Thanks, Kara. And thanks so much to all of the folks who have shown up, and particularly to all the folks who are and the organizations who are involved in sponsoring this event. To provide some background and some history, since 2016, a coalition of community partners in the Lower Cape Fear River have been deeply concerned with and focused on trying to understand the types of chemical contaminants that are in fish that are found in the Cape Fear River that are consumed by people in the Lower Cape Fear River. This was spurred in part by the discovery of Gen X, that was a PFAS compound that was produced by the Chemours facility outside of Fayetteville, North Carolina, and which has spread throughout the Lower Cape Fear region in North Carolina. This has spurred, again, a much broader interest in and concern with the effects, both environmental and on human health, of PFAS more broadly, not only in the Lower Cape Fear River, but more and more as we're discovering sources and the spread and dissemination of PFAS throughout our environment, throughout North Carolina, throughout the United States. This is not just in waterways. This is not just isolated incidents. It is really in every facet of our life and in every part of our environment. So as we have been focused on this, we really have been concerned primarily with looking at the ways in which people are ingesting PFAS in the fish that they're consuming out of contaminated rivers, but more broadly, and what the topic of today will be, is really thinking about how it gets into the food that people hunt, that people collect, that people garden and produce in their own homes. This is a serious concern right now, of course, because there is such a dire problem right now in the United States, and particularly in parts of North Carolina, with food security. And as people become less and less food secure, they will be, of course, going to sources of food that are free, right? That are we don't have to go to the grocery store and pay for. And if those sources of food are contaminated with PFAS, we also have to be concerned about the immediate and long-term health consequences. So I'm so entirely grateful for these experts who have chosen and are willing to join us and share their knowledge and wisdom on this topic today. It is entirely relevant, it's entirely needed, and the people who are listening today will be very grateful for all that you have to teach and all that we have to learn. Thank you so much for being here.
Great. So we have a great lineup of experts today. We're going to hear from each of them and then answer all questions at the end. So as each speaker is talking, feel free to be typing as many questions as you want using the Q&A feature, and then we'll get to all of them at the end. So today we'll hear from Dr. Courtney Carignan, Dr. Pingping Meng, and Dr. Lipeng Feng to hear three different perspectives on PFAS in our food systems and potential health consequences. Our first speaker is Dr. Courtney Carignan. She's an exposure scientist and an environmental epidemiologist at Michigan State University. She conducts biomonitoring and health studies for all different populations, and her research has contributed to public health interventions that are aimed at reducing human exposures to flame retardants, PFAS, and arsenic. So with that, take it away, Courtney.
Thank you. Thank you so much for having me. I am an associate professor now. I just got tenure. So I'm still working on updating everything. I just noticed it on that last slide. Okay. Can you guys see my slides? Okay. Pretty good. Can you see it?
Yep, looks great.
Okay, great. So my goal over the next 10 minutes is to help you visualize how PFAS transfer and accumulate up the food chain within agricultural settings. I'm going to do that with some telling some stories and also sharing some research results. So in 2016, it was discovered that there were elevated PFAS in groundwater at Stone Ridge Farm in Arundel, Maine, and subsequently in milk from the dairy. It was a small dairy operation. And the anticipated source of the PFAS contamination was sludge and paper mill waste used as fertilizer on the pasture. I'm going to talk a lot about sludge and biosolids. So I just want to sort of explain a little bit what that is. So wastewater treatment plants generate a solid waste we call biosolids, and those are created from sewage sludge. The most common management option for biosolids is to beneficially reuse them as an agricultural amendment. But because of the risk of pathogen exposure, often there is a pathogen reduction before biosolids reuse. Right, so you're going from, let's see, I'll show you on the next slide. All right, so our homes, when you use the facilities, or you are using or doing laundry or using a dishwasher or using your sink, you're creating wastewater that then goes underneath the ground through the pipes and ends up at a wastewater treatment plant. The same thing happens for industrial operations, but they also have something called effluent, which is often the wastewater from those operations. let's see, a little animation, which is just showing you that waste from residences, actually, and industry, go to landfills, and landfills have a juice that comes out of them. We call landfill leachate, and that leachate also goes to wastewater treatment plants. So there are a lot of different routes that of inputs into wastewater treatment plants. We call that often the influent into wastewater treatment plants, and then the effluent is the liquid part that often goes into a water body after it's been treated. And there's also a solid part, and that solid part is what we call biosolids. And those usually go through treatment, like I mentioned before, to remove pathogens before they are land applied. So the majority of biosolids of that solid waste are land applied. I think it's around a third or a bit more. The other is either incinerated or landfilled. And once it's on the farm, it can be taken up into crops, into livestock, and also the PFAS can migrate through the soil into groundwater, and you can get runoff into nearby water bodies. Right. So we know that the presence of PFAS are well documented in wastewater treatment plant effluent, and biosolids. These have focused primarily on the perfluoroalkyl acids, which are a small fraction of the thousands of PFAS that are known to exist. A recent study found that the total PFAS concentrations before and after treatment were dominated by the PFAA precursors, and particularly the DIPAPs. And this is indicating a shift from the predominance of PFOS, which I'm going to talk about probably a little bit more in a second. They also found that heat treatment and composting of raw biosolids were found to transform the precursors into the PFAAs, and they indicate that the precursors are likely underrepresented by other studies and make up a larger percentage of the total PFAS concentration in biosolids than what was previously estimated. So it's a bit complicated, and 10 minutes is not really enough time to explain, but I'm going to do my best. So this is showing precursor transformation. So you've got transformation from this precursor ET fossa into PFOSA and into PFOS. You can also get transformation into PFHXS and PFBS. And so we know that PFAS can be taken up into plants, and that they're taken up into plants and crops more if they have a shorter chain length. So like PFBS has a shorter chain length, for example, than PFOA and PFOS, which have a longer chain length of 8 carbons. And we know that they will accumulate more in animals if they have a longer chain length. So there's sort of a little bit of a trade-off, I guess, in terms of accumulation between short chains and long chains. And this pathway that we're interested in, and we're talking about today, is from the farms, like the farmland, into the crops, into the livestock, and into the humans. So this is primarily what we're going to be talking about. So in Maine, after the contamination at the Stone Ridge Farm was discovered, a map was released that showed all the biosolid application locations in the state, and the state began testing these locations, and they started finding PFAS at more farms. So one of the farms where they found really a notably high level was at Songbird Farm in Unity, Maine. Concentration of 8,000 parts per trillion in well water, which is quite high, and 475 parts per billion in soil. And this was devastating, obviously, to the farm. They basically lost their business. And while it was devastating, at the same time, one of the things that the father here, Adam Nordell, says is that it was also a blessing in a way, because the person that they bought the farm from actually died of pancreatic cancer. And he feels that, this discovery likely has helped protect his family from a similar fate. So in Michigan, around that same time, began a PFAS initiative for the state industrial pretreatment program. where they had the treatment works identify sources of PFOS into their collection systems. And by 2020, many of the systems had achieved substantial reductions by working with their most significant sources. And so in Michigan, one of the significant sources is metal plating, because PFAS are used as a fume suppressant in chrome plating, peroxide chromium. And then that ends up in the industrial effluent that goes to wastewater treatment plants. And the PFAS are not removed during that treatment, and so they pass through, like I kind of talked about before. In Michigan, they also tried to find, where that contaminated biosolids had been spread. So one place was in Lapeer, Michigan. They tested the soils and found elevated levels of PFOS at 496 parts per billion. And this field typically had a corn, soy, wheat rotation. It's since been converted into, I believe, Christmas tree farm. So we were able to get samples from that field, and we did a study where we created a model to estimate exposure from the soil to the crop to the cattle to the people, to a farm family. So the model that we did was in particular for a farm family, similar to the EPA biosolids risk assessment that was done a couple years ago. We did have some good news, which is always nice in PFAS. We didn't find any detectable PFAS in the corn kernels, and Pingping helped with that. She actually identified sort of some interference that was giving some false positives in kernels. And so if you're, eating sweet corn, you can feel pretty good that there probably isn't any PFAS in your sweet corn. The stover, which is the other plant part, did have PFAS in it, and the stover is eaten by cattle. And so that's why this model is really focusing on this pathway through cattle. So we had paired samples. Have I been going for 19 minutes, or is it counting down? I'm at 10 minutes.
You're close to 10 minutes, so if you could wrap up in the next 5, that would be amazing.
Okay.
I know,
so we had
it's a
paired
lot
samples.
to get through.
I'm looking at my timer. It doesn't make any sense. So we had paired samples, and you can see that the transfer of PFOS from the soil to the stover. And from that, we were able to calculate accumulation factors, which were similar to accumulation factors that had been from previous studies, at least compared to the median from those studies. So a lot of those studies had a really large range. And we also saw the lower uptake into PFOS and PFOA compared to the shorter chain PFAS like PFBA, which has 3 carbons. We had a dynamic pharmacokinetic model for cattle over the first 2 years of life, and we can see higher levels in liver, followed by serum, kidney, and the lowest in muscle. And then we use those concentrations to estimate exposure for children, a child, and an adult who were exclusively all of the meat that they were eating was coming from the cattle, these contaminated cattle. And we can see higher exposures for a child compared to an adult, which is pretty typical in most dietary models, and that we have lower exposures from eating the muscle compared to eating the liver. And we did a model where we actually modeled PFOS exposure across concentrations in soil, and then extrapolated from there, sort of where maybe a screening value might be, where the level was similar to the reference dose from EPA for PFOS. So we did that for soil, for stover, and then also for beef. So here we can see that concentration that you might use as a screening value based on this model is around 0.11 for a child. So here in Michigan, Grosta Cattle Farm was identified in January 2022 as a farm that likely had elevated PFOS in the cattle meat, and they did find up to 2.5 parts per billion in meat. So they stopped selling their meat and is now working with MSU in some research. And we also have a study where we looked at a paper mill. I don't think I have time to get into it, but this is another pathway of paper sludge being dumped. There's also other reports and examples of where paper sludge has been directly applied to farm fields in some places. And so we have a biomonitoring study where we've really collected a lot of samples and a lot of data on these exposures through drinking water, diet, and indoor environment. And we can take concentrations in drinking water and estimate exposure, which we can compare to reference doses. And we can see much higher exposure before the drinking water intervention compared to after, but we see a real change in the PFAS that are present. We can do the same for eggs and venison and beef. And we can see that in a risk assessment, we have hazard indices that exceed 1, indicating potential risk for some of these products. We found that the home gardens had relatively low PFAS in produce, probably a lot lower than when people were irrigating with their contaminated water. The water really seemed to make a big difference for uptake from irrigation. And then we also had some results from site and background farms, and you can see that even though the levels in spinach were relatively high and had a marker of the site, for the child, the levels were relatively low. And even for adults, because people don't actually eat that much spinach on average, whereas like for children, the young children tend to eat more squash, for example, and so you can see those were higher. I'm going to skip right through that. And there's also examples of this with aqueous film forming foams, which have been used all over the place and can move into groundwater and be taken up into wells, including agricultural wells. And there's some examples of farms in Colorado. This was an organic farm that had to get shut down. And in North New Mexico, this farm actually was a cattle, a dairy, and he had to euthanize, 4,000 cows. It's a really devastating situation for him. So this is not really that new. Tenant Farm, if you guys have watched Dark Waters and knew the story about DuPont, this kind of went back to the 1980s and 1990s. So this discovery of Rob Bilott, played by Mark Ruffalo in the movie, started in around the late 1990s. And in Decatur, Alabama, there's some evidence that 3M knew, dozens of years ago about contamination coming from their facility in Decatur, Alabama, into the wastewater treatment plant and the biosolids that were land applied. So thank you, and I'll pass it on to Pingping.
Thanks, Courtney. That was great, and it's going to flow so nicely as we hear from Dr. Pingping Meng, who's an environmental chemist at East Carolina University here in North Carolina. Her research focuses on the occurrence, transport, human exposure, and remediation of emerging contaminants in the environment, like PFAS. And this includes studying PFAS uptake in homegrown foods in communities around Fayetteville, North Carolina.
Thank you for having me today. So can I move my slides here, or should I say next?
Just say next.
If you want to, just say next slide and I'll advance.
Oh, okay. Yeah, thank you for having me here today. So today I'm going to talk about PFAS contamination beyond the tap. So we are going to talk about PFAS in the garden here. So this research was done when I was a postdoc at NC State working with Dr. Detlef Knappe, and the project was led by Dr. Christopher Higgins at Colorado School of Mines. And Dr. Owen Duckworth did a lot of work helping us to connect with the communities, getting the samples and report all the data back to them. Next. So like we all know, before. So comparing to drinking water, the importance of PFAS human exposure through the food remains poorly understood because water is relatively easy for analysis. And there's an important question is like, how important is PFAS exposure through drinking water comparing to that through the food? Next. And particularly in North Carolina, different from Maine and Michigan, we are facing the challenges from a novel type of PFAS called the per and polyfluoroalkyl ester acids, or the PFEAs. And the Chemours facility have been discharging those PFEAs into the environment either through wastewater discharge or air emissions. And close to that, for chemical manufacture, many people have been harvesting and consuming fruits and vegetables from their own garden. But the uptake of those PFEAs into local produce still remains unclear. So we really want to answer the question whether for the community, like is garden produce an important route for PFAS exposure for them? Next. So this is a map. To answer that question, we were very lucky to have 5 residential gardens volunteered by the communities enrolled in this study. So this is a map of that impacted the community. This is a figure from the NCDEQ website showing the PFAS levels in their groundwater, and each point here represents a groundwater well. And the brown zone represents the Chemours property boundary. So though those 5 sites are within a 6-mile radius of that Chemours plant, and in total we have got 53 fruits and vegetables from the 5 residential gardens, and the produce were harvested between 2013 and 2019. So also among the 5 sites here, next. This is the produce inventory we made. In total, there are 53, and we grouped them into 4 categories, like water-rich samples such as berry, squash, or tomatoes. And we also have tree fruits like apples, pears, oil-rich samples such as 2 pecan samples, and starch-rich samples such as sweet potato, potatoes. From the 4 sites, A, B, C, D, and E. Site E is a site we picked as a control site because it has relatively cleaner groundwater. It's one of the green dot in the previous slides, which means their groundwater has non-detectable levels of PFAS, or neither of the PFAS is above 10 nanogram per liter. Next. So next, I'd like to first show some general PFAS levels in those 53 produce. So among those 53 produce, there are 10 PFAS that were detected in more than 10% of the produce. And the pie charts show the detection ratio of different PFAS. Among the 10 PFAS, 8 of them are actually those novel PFAS, or fluoroethers. And as you may see, one of the PFAS, PFO2HXA here, it was detected in all 100% or all of the 53 samples. When we look at the mean concentration of different PFAS, you may see the mean concentration of fluoroethers are orders of magnitudes higher than the non-ethers, the PFEAs or PFHAs, suggesting those ethers, the novel PFAS, are the dominating PFAS in the PFAS signature in those produce. Next. Another thing we'd like to look at is our PFAS types matters when we talk about PFAS levels in edible parts of those plants. And we do see the water-rich samples such as blueberries, blackberries, which are shown in red color in this figure here, shows the highest level of PFAS in the produce, while the starch-rich, which is shown in green color here, has the lowest level of PFAS detected. That suggests the type of plant does have an impact on PFAS levels in plant uptake. Next. And then it comes to our question is we want to understand or compare how important is exposure from food through the water, through the food compared to that exposure from drinking water. Next. To answer this question, we made an assumption, like we assumed the water that residents are drinking were containing 10 nanogram per liter of GenX, which is the EPA regulation and also our state guideline here. And suppose a kid between 3 to 6 years will drink, say, around 1.5 glasses of water per day, while an adult may drink around 4.5 glasses of water per day. Next. And this figure shows how much of food they can consume from different sites to reach equivalent amount of GenX exposure through the water they were drinking. So this figure here, the left figure, shows the kids how much the produce they can eat from 3 different sites, site A, B, and E. And like we said, site E is a relatively cleaner site, so the levels of PFAS in the produce are much lower than for the produce from site A and B. And the dashed line here is showing the recommended produce intake by the EPA exposure handbook, which for kids would be around 180 grams per day. And as you may see, if they were consuming the produce from site A and B, the amount they can consume to reach equivalent amount of GenX exposure from drinking water is only 22 or 17 grams per day. And for the adult, the results are very similar, which means if you are consuming produce from the site A and B, which are more impacted sites, the amount would be very small comparing to their recommended produce. While for site E, the site is relatively cleaner, then it's safe to consume the produce. So, and just to show how much of, say, 22 or 17 gram of produce looks like, we show a photo of here how much 1 ounce of blueberry may look like, and it's only one handful. So I guess based on these results, next, we can say in impacted communities, the uptake through residential garden produce may be a very important route of PFAS exposure in addition to drinking water here. So now that we know those produce are impacted by PFAS, next, what we wonder is there anything we can do to help minimize PFAS plant uptake? So this is a study done by two previous postdocs at NC State, Dr. Lee and Dr. Zhi with Dr. Owen Duckworth. So they did a greenhouse study with PFAS added to the soil, and they want to see how much PFAS can be moved from the soil into the plant. And this figure shows they detected PFAS in the lettuce leaves, and all of the 8 PFAS they studied, they were found in lettuce leaves. The y-axis scale shows the PFAS concentration in the lettuce leaves, and you may see the number that's extremely high. That's because they have dried the lettuce leaves before extracting them. So that's nanogram per gram in dry weight. And another thing they find is adding clean compost did lower PFAS concentration in the lettuce leaves, because if you compare the 0% compost data, which is in white bar here, that's the control study, and comparing it with increasing levels of compost up to 20%, which is in the blue bar here, you may see a substantial decrease in the PFAS levels detected in lettuce leaves. So we say adding those clean compost did reduce PFAS uptake, but we also recommend you need to analyze the compost for PFAS before use, because there might be compost on the market that was derived from biosolids. And like what Courtney just told us, many of the biosolids may be enriched with PFAS in them originally. Next. So our work hasn't stopped there. So more work is on the way, because now our focus has been moved to the importance of ultra shorter chain PFAS. And many of you might have already seen this study done by NC State researchers. They find high levels of short chain PFAS in Wilmington residents' blood. So Wilmington is a city downstream of the Commerce facility. And before this genetics problem was revealed, residents have been drinking impacted water for a long time. And the concentration, for example, for one of the ultra short chain PFAS TFA wants to be as high as 108,000 nanogram per liter in the drinking water. And correspondingly, exposure to those high concentrations of ultra short chain PFAS may contribute to their dominance in the human serum. For example, the TFA concentration is as high as 17 nanogram per milliliter in the human serum here, even though those ultra short chain PFAS has been a long time considered as non-bioaccumulative. Next. So our question is, would the diet also contribute to ultra short chain PFAS bioaccumulation? While in the case of Wilmington, drinking water might be their dominant exposure pathway. Next. So to answer that question, our lab at ECU has started a pilot study, which is we collected samples from grocery store in Greenville, North Carolina. Though the produce, we don't have much information about the source. Therefore, we just regarded them as market samples. You may be able to get them from other chain grocery store outside of North Carolina. That's very likely. And we optimized our extraction and matrix cleanup method as well as our LCMS methods, because ultra shorter chain methods are extremely challenging due to the lack of analytical methods, particularly in complicated matrices. And here are our preliminary data. Next. So among the 3 produce, we analyzed tomato, squash, and sweet potato, and one ultra shorter chain PFAS, the trifluoroacetic acid, TFA. The structure you may see is extremely short chain PFAS. We detected high levels of TFA in those 3 market samples. If you look at their Y scale, the concentration may be as high as 100 nanogram per gram, and that's wet weight. So we did not dry those samples. And if you remember the previous slides, we showed the level of the other ethers in the samples maybe between 0.02 to 2 nanogram per gram wet weight. So the concentration of this TFA is orders of magnitudes higher than the other PFAS. And another thing that stood out is different from the other PFAS when we find the water-rich samples actually have higher level of PFAS than the starch-rich samples. In this case, at least we were seeing comparable levels of TFAs in those 3 samples, regardless of its produced type. So this is very preliminary, so we are still working on getting this method finalized next. And the plan is to measure ultra-shoulder chain PFAS in the samples we get from the impacted community, which are the 53 samples we have, and we also collected a few other samples in the following years. Next. So to wrap up, we find elevated levels of PFEAs were detected in those residential garden produce near fluorochemical manufacturers in North Carolina. And we also find those levels in produce vary by plant type, even though the ultra-shoulder chain may behave differently. More study is on the way. And with water-rich produce exhibiting the highest level. And we think consumption of those residential garden produce may represent a significant exposure pathway in impacted communities in addition to drinking water. And additionally, we find clean compost can help mitigate PFAS uptake by plants, but the use might be applied with care. Finally, we just started the ultra-shoulder chain work, and based on the preliminary data, they may dominate in PFAS signature in produce samples. Thank you.
Thanks so much, Pingping, although that's kind of depressing. So one of the reasons, right, we talk about this and we worry about PFAS exposure, a lot of us are thinking about the little ones that drink water or eat potentially contaminated foods. We think about our families and not just our own exposure. So last but not least, we're going to hear from Dr. Lipeng Feng. She's a medical doctor and a researcher at Duke University. Her work has focused on understanding the mechanisms of pregnancy complications associated with placental development. And this translates to then the clinical care setting by helping to identify risk factors and biomarkers for early prediction and ultimately prevention of adverse birth outcomes. Thank you, Dr. Feng.
Thank you for the introduction and good afternoon, everyone. Next. I usually advocate for water filters, so I have to claim no conflict of interest. And we heard enough bad news today, but I have to deliver another bad news. Sadly and alarmingly, PFAS exposure doesn't begin after birth. So we were talking about kids and adults, but it starts in the womb. Babies are exposed to PFAS even before they cry for the first time because these chemicals can cross the placenta during pregnancy and reach to the developing fetus. Recently, we found that PFAS can also accumulate not only in the cow milk, it's also accumulated in breast milk. In utero In utero PFAS exposure sets the stage for a lifetime of increased disease susceptibility, such as chronic diseases relevant to metabolic, endocrine disrupting, neurodevelopmental toxicities, and lastly, cancer. It is also called the developmental origin of health and disease. So this is why my lab has been focused on understanding how perinatal and perinatal PFAS exposure affects our health. Specifically, my lab pioneer studies using PFAS mixtures and an emerging PFAS compound, PFBS. There are numerous PFAS toxicity studies out there, but Leslie focused on PFAS mixture, but evidently we exposed to PFAS as a group of chemical compounds. And Pingping already talked about the short-chain PFAS. PFBS, when I start interested in this short-chain PFAS, there's no toxicology studies available for us to determine the toxicity of this compound. So my lab initiated the study. Over the years, we found many toxicity effects of this compound, and evidently it was not detectable in North Carolina drinking water, but now is the number 4 frequently detectable PFAS compounds in our drinking water. Furthermore, my studies are highly relevant. We can skip this if you're interested in them. regulations, we can talk about this. And if you ask questions, we can talk about this, and we can all skip this slides over. And oh my God, I didn't know we actually on this slides. You can move forward too. Yeah, this is the PFAS and PFAS mixture and PFBS I was talking about. So furthermore, my study are highly relevant to North Carolinians by using a PFAS mixture that mimic PFAS levels in Pittsboro and sea drinking water. You can move forward. Next, which is highest in the RTP area. It's the same water system from Wilmington. We talked about Wilmington earlier. You can see the total level is 760 ppt. Courtney talked about the farms was 475 ppt. You think that's high? Pittsburgh drinking water is even higher, which is really scary. And the PFBS levels used in my lab also mimicking the drinking water levels in also mimicking PFAS levels in this drinking water level. I highlighted in yellow. Move forward. Next. Now, what did we find? Overall, our findings focused on maternal health outcomes, fetal offspring development, and mechanistic studies through placental development and function. Maternal health outcomes includes that I'm giving you 10 minutes talk, but this is years of work. We found PFAS exposure increased risk with gestational hypertension, diabetes, endocrine disruption, immunotoxicity, cardiac function, and postpartum depression, which sound depressing as well. Next. So what do these findings mean to you? Oops, next. Yeah. So first, gestational hypertension and preeclampsia can lead to organ damage or death in moms. For babies, they could have, next, lower birth weight, in utero growth restriction, preterm birth, neonatal complications, and stillbirth and perinatal death. This is, okay, you can go ahead. This is only the beginning. Long-term impacts of this pregnancy complications includes in moms, increased risk of cardiovascular disease, higher likelihood of hypertension and diabetes. In the babies, the fetus, the children suffers from can suffer from preterm birth relevant long-term outcomes, also increased risk of hypertension, heart diseases, and metabolic disorders. Next. For immunotoxicity, we found that, next. For immunotoxicity, we found that maternal PFAS exposure decreased the immune response to Tdap vaccination and caused B cell dysfunction in mothers. So B cell function means it can cause increased disease burden. Next. Next. You cannot see it, such as cancer, because our immune system defends our body from cancer, and infectious disease, obviously, and reduce vaccine effectiveness, potentially elevate outbreak risk and increase economic burden, as you can, we just experienced a pandemic. So you understand all this public health impacts. Next. Oh, this one. And yeah, our study also found maternal PFAS exposure to that environmental relevant PFAS mixture led to postpartum depression in mice. And the impact of postpartum depression, first of all, is very high. 1 in 7 mothers suffer from postpartum depression. And it is sadly and shamefully is the leading cause of a maternal death in our society right now. It causes higher risk of chronic conditions due to stress and impaired bonding and caregiving, impacts on infant development, family social impacts, and family functioning, and long-term child outcomes. Speaking about long-term child outcomes, now we're transitioning to how PFAS exposure during pregnancy affecting offspring's health. Next. Next. You can go next. And next. Yeah, normally the pregnancy mass is very hypoxic compared to other organs. The hypoxic condition is to protect the fetus and embryo from oxidative stress, maintaining the cell stamina and promote angiogenesis, like vessel development, which is important for the placenta and the baby. We reported that maternal exposure to the PFAS mixture, the same mixture, led to hyperoxygenation and oxidative stress in the placenta and embryo. Our preliminary data suggests that this may be partially due to altered mitochondrial function induced by PFAS exposure, leading to oxygen accumulation. And indeed, next, we found that maternal PFAS exposure disrupted mitochondrial function. Disrupted the mitochondrial function. If you learn biology in high school, you will know mitochondria are energy fuels of our cells. And this is particularly in the fetal brain, specifically in the cerebellum, and cause oxidative stress and developmental delays in this brain region. You can see the staining of the cells in this brain region. And this ultimately leads to changes in locomotor developmental trajectories in the fetal sex-dependent manner, with female offsprings being particularly vulnerable in this case. Next. PFAS chemicals are surfactants if you understand the chemical structure. So if you are drinking surfactants, you will feel it in your gut. So not surprisingly, both PFAS and PFBS exposure disrupted gut barrier homeostasis. In general terms, is you might have a leaky gut and increase the risk of intestinal inflammation in rat offspring. We further identified PFAS exposure as disrupting gut-liver crosstalk. Next, I'm actually talking about the next slide. Leading to intestinal health disturbances in rat offspring where the gut microbiome. So if you drink surfactants, it affects your gut microbiome. That's not surprising. And this is all the healthy facts I'm telling you. And what are we going to do about it? So next. Avoid products containing PFAS, which is really hard. Like it was new to me from the last two talks, our farms, food, and not only drinking water. I always advocate for using water filters during pregnancy to effectively remove PFAS. The regular water filter doesn't work. You have to use reverse osmosis filters. Some families actually install the whole reverse osmosis filter system for the whole water system in their household. That might also mitigates your own garden contaminations as well. And of course, we need to support regulation to limit PFAS productions and emissions. That will be the ultimate solution. Otherwise, we still have to drink. We still have to eat our vegetables and cow meats. And the next. So take home message from me. PFAS exposure can harm mothers during pregnancy, and even small amount of PFAS matters. We need stronger rules to limit PFAS and mitigating PFAS exposure in daily life, especially during pregnancy and infancy. Thank you.
Thank you for bumming us out some more. So we already have a few questions in the Q&A. And I know I said this during our last webinar, but I'm going to say it again. I'm a social scientist, which means I did not understand quite a few of the things each of our very smart panelists brought up. And I'm going to ask some questions during this discussion that might seem really simple or basic to some of the scientists and grad students in this webinar. But all that to say, there's no dumb questions, there's nothing too basic, and there's nothing too nitty gritty. Like, we really want this to be a space where we can take advantage of having these really smart women on Zoom with us. So don't be shy using the Q&A feature to ask any little or big question you might have. But I'm going to exert my authority to ask the first question. So Lipeng just mentioned this idea of, like, in a perfect world, we would not use PFAS, they wouldn't be in our environment and our products, and we could avoid exposure. And we know we are not there yet. So when we think about potential exposure to PFAS through our foods, and we know that PFAS are everywhere, who do we think are most at risk? Is everybody growing fruits and veggies in their garden at the same level of risk for potential PFAS exposure, or who do we think should be most concerned or most thinking about this issue?
I would say for people living close to, like, say, major source of contamination, maybe at higher risk. So like we said, the study we did was like within a 6 mile radius of a very large fluorochemical manufacturer. So I would say this is a case study. It does not say every home should give up their, like, residential garden plan.
I would also see professions. What kind of professions? Living near our veterans and military soldiers are high risk to PFAS exposure, and firefighters. Unfortunately, they're all heroes to us, but they're more exposed to PFAS. And I agree with Pingping. It depends on where you live. And well water. So far, I don't. It's just my knowledge, and Pingping and Courtney can confirm well waters has low levels of PFAS, at least in our RTP area.
Yeah, and private well owners need to test their own wells. So by the end of this year, I think all large public water systems in the US should have been tested, even though not all states have enforceable drinking water standards. So probably see differences in treatment and action across states. But private wells are sort of the next frontier in terms of where to push to get testing and help people get access to testing, because it's hard to know where you'll have sort of these hotspots because of how AFFF has been used and not tracked for decades. And so people with private wells, really should be testing. Cyclopure, I think, is a relatively inexpensive test that works pretty well. And then a lot of state labs can do testing or maybe even offer it for free. So,
yeah, that actually relates well to one of the questions we got from a nonprofit community-based organization here in North Carolina called Toxic Free NC. And so they were asking, do we see lower levels of PFAS in well water? Is it safer? And is there a way for farmers to test their well water? And no would be the answer. And I don't know what it's like in Michigan, Courtney, or do you know of other places in the country where there are, like, services for farmers that want to test their water, or is it really just on a consumer by consumer basis using something like Cyclopure?
I think if anyone's doing it, it would be Maine. Maine has been doing a lot on this topic, and particularly with relation to farms. But I don't know offhand if they have a program, like what that program is and how it's funded and what it looks like. But they have in Maine gotten funding to support PFAS testing and action in agriculture. And so if anybody has it, I would think it would be Maine. But yeah, I mean, I think that one of the big, the elephant in the room with this topic is that it's very difficult to test for a contaminant if you don't have a good safety net in place for these farms. Because if you're going to put a farm, if you're going to test a farm, it's going to put them out of business and it's going to bankrupt them. Farms aren't going to want to test. And so largely that's what we've been seeing And Maine also has a safety net for farmers, not really through the government initially. It was through an NGO, through a foundation. I think that that's part of, maybe what's helped this issue advance in Maine more so than other states.
Yeah, someone wanted to follow up on that and was wondering, is the biosolids example from Maine that you gave the tip of the iceberg, given that other states aren't testing yet? Or is Maine an example of a worst case scenario? Because in North Carolina, biosolid application is everywhere, in all caps, but virtually no testing of the wells.
I don't think that Maine is unique in this issue. I think the only way Maine is unique in this issue is that they're testing and taking action. You would like this. There was a really nice social science study on how do we communicate about this. And they found that by talking of, sort of prefacing it with Maine is looking and therefore, taking action, you would expect that Maine foods would be safer, essentially because they are testing and taking action to sort of counter this perception that that there's more PFAS agricultural contamination in Maine. Actually, over time, we're probably seeing less, I would expect, agricultural contamination in Maine because of all the things that they're doing, sort of similar to how Michigan has been doing a lot of testing of PFAS and sort of lights up on a map because we have so many test locations. But that's because Michigan's been looking. And so I don't think we have hard data, or at least I don't, on sort of comparison of contamination between states. And it's hard to compare when you have different approaches being used. You really need a more equitable baseline to sort of compare between.
Yeah. Lipeng.
I collaborate with RTI. I was hoping EPA, it's local, can have a lab and open to public. So I talked to RTI people about this. I said, why don't you open your lab to public? And it's mainly legal consideration. For example, I'll give you an example. As a physician scientist, I care about my patients' questions. So if I told them this PFAS accumulate in breast milk, they will ask me, should I let my babies drink formulas? I don't even know how to answer that. So I asked my collaborators, I said, why don't you measure PFAS levels in the formula across the brands to identify the least contaminated? They were like, then the company is gonna chase after us forever if they report this. So nobody wants to take the legal responsibility for that. They will ask you to validate the data this way, that way, this way. They may sue you, and it's just such a mess.
So relatedly, we got a couple more exposure questions, and then we need to dig in, Lipeng, I think, to PFAS once it's in the body. So, I think one thing from Pingping from your slides that was really helpful is if we use compost, we can reduce the amount of PFAS in the soil and ultimately the amount of PFAS that gets into the food we eat. But is there an easy or cheap way for people to get their soils tested, whether it's like a farmer on perhaps a larger scale or me growing things in my backyard garden? Are there resources available for people that you know about?
Yeah, I think that's the thing that's not so good is for when we talk about PFAS, the analytical cost is always going to be a huge burden, right? And like for example, I think I have heard of the cost of sending a PFAS to a commercial company for PFAS analysis. Of course, they have all the verification and all those things. And using EPA method 1633, the cost may be above 500 or 800 per sample. So I think that's a huge cost, economic burden for a farmer that to get and also how many sample you plan to send, right?
Right.
Like if you have a large yard, you can't just pick a soil from one corner and yeah. So that's I think there needs to be a way, I guess, either like the state of Maine did a great example, like the state is taking a lot of responsibility providing analytical support, but to the best of my knowledge, I don't know whether there are resources that the farmer can apply for free for testing. Yeah, so sorry about that.
Womp womp. Well, but there's been like 70 some people on this webinar today. And so perhaps there is a brain trust that can form of how can we replicate some of the things that have worked really well in places like Maine to do something in North Carolina, especially now that we have science that shows us like there are things we can do to help reduce exposure. We just need the resources to be able to do them. Um, Similarly, like I assume the same thing applies to testing the actual fruits and veggies themselves. Like you would need a commercial lab to do it. Is that right, Ping?
Yes, and I think for those more legacy PFAS, the reason why we had to develop the method in our lab and analyze those samples for fluor ethers is if you send something to a commercial lab, most likely they're going to focus on the PFAS that's more like kind of studied, like the legacy PFAS, PFOA, PFOS, that's for sure, but also the other maybe around 40 PFAS that's included in the EPA method 1633. If you come to them to say like we want to do the ultra shorter chain or one of the ether PFAS that's not included in that method, most likely the commercial lab is going to come back and say we need to either a lot of money to develop a new method, or we will not be able to provide reliable results. So that's going to be a case by case scenario. So,
Okay. So now let's assume that we've been unable to avoid exposure, or maybe we just want to be really careful. What can people do? One, to find out if they have PFAS in their bodies, and two, if they know they have PFAS in their blood, is there anything they can do to reduce the levels of PFAS in their bodies? I think this is probably a Lipeng and a Courtney question. So whoever wants to go first.
Sorry, I
So
just typed the answer.
yeah,
That's okay.
Go
We
ahead.
can have it both places.
for women, we actually has a way of naturally remove PFAS from our blood is menstrual cycles. So no better ways. I heard people use giving blood to clean their blood. It's terrible.
Someone mentioned, and I know I've heard it in the past. I don't know, Courtney, if you can speak to it, that cholesterol medicine. What is the scientific consensus on that these days?
Polystyramine is getting 2 thumbs up. It's much better for PFOS than for PFOA elimination. It does have some side effects like flatulence. Um, Doctors I know are prescribing it for people with levels over 50 something parts per billion in their blood of the sum of the seven MESM, PFAS, and are seeing really good results with that. So we have a session on it at the National PFAS Conference coming up in June that I'm co-chairing. Really excited about it. It's something people in affected communities have been asking for a really long time. And I'm really happy to see this moving forward. Other things that work are phlebotomy, so serial phlebotomy. There's some studies on that showing like a 30% reduction particularly with donation of plasma over serum. And then I've also heard anecdotally that for profit, so if you, not for profit, but if you go to a place that does a plasma donation and will pay you to do it, that they actually may be filtering the blood that would remove PFAS. So I was interested to hear that. Katie, I can introduce you to the person who told me that at the
Wow.
conference, because I know this will be interesting to people. And trying to think, am I missing any? There's some interest there's been some things around like microbiome that, I don't know if anyone's seen these like probiotic supplements that maybe could reduce PFAS, but really haven't been shown to do so in humans. And
So it sounds like where the science is right now is with the cholesterol medicine and donating blood, but there may be other options soon. We just don't have as much
oh,
of research on it.
fiber, that's the other one, is the fiber supplement. So there's a protocol with like Metamucil that looks promising,
Hmm.
um, but might not again be tolerated by everyone.
All right.
So I could take lots of Metamucil and I'd be good, but my husband would not. Too much information for me.
Yeah, I
All
think
right, now we're going to get into some of the nitty or grittier methodological questions. Okay, so starting with Lipeng, can you talk a little bit more about what models you used, whether it was cell culture or mouse models?
yes, mouse model. My lab is like a little zoo. For the immunotoxicity study, we use rabbits because rabbits resembles the immune system in human. And for other studies, we either use rats or mice. The reason for that is the placentas are similar to human placenta. The human placenta is just a very unique organ. None other species are even close to human placenta besides rodents and non-human primates. We also use human cellular model, the mitochondrial function studies and placental biological studies. It's also replicated in human cellular models. And I didn't talk about my epidemiology study. We also have my lab is a spectrum of human study, mice study to cellular model. The hypertension, the hypertension studies is all across all spectrum. Human study, mouse model, and cellular model. Immunotoxicity of PFAS is not a mystery. It's the first toxicity studies show. The B cell dysfunction is novel, what we found.
So all three of your work touches on this a bit, and we had a question in the Q&A related to the difference between adult and children exposure to PFAS. And so I know we saw on some of the graphs the difference in the number of blueberries that an adult could eat versus a child could eat before crossing the threshold of allowable Gen X exposure. So is that because they're just smaller than adults? Is there something physiologically different? Can you talk more about how we think about the difference between adult and child exposure to PFAS?
Do you want me to talk about it, or you want to Pingping talk about the blueberry?
You can all talk, whoever wants to go first.
I think the reason why children are like more vulnerable, let's say, like considering how much food they can eat to get the equivalent level of genetic exposure through drinking water is like what Katie said. They are smaller, so the amount, of course, they are gonna be the limit. It's easier for them to get to the limit. But I think also because children are at a developing stage, so I'm not an expert there, but I feel like because children are a lot of their systems are developing, though they might be more vulnerable to PFAS comparing to, say, an adult, like your system are more fully developed. But I guess Dr. Feng would have better answers for this.
That's exactly my point as well. I call it developmental origin of diseases. In my culture, Pingping probably has, this, we were born 1 year old. So what is happening in your mother's body and while you're growing in your mother's body, it's important after your birth. So whatever is programmed during pregnancy of impacting your long-term health, you were born who you are. And that's what I always see. And especially during fetal development, your organs are forming, especially I study the brain, the fetal brain. Your fetal brains are very plastic, which means it can be molded into this way, it can be molded into that way. And any disturbance from the environment, from your mom's health conditions may program your brain in a different way. It might be good, it might be bad, but for PFAS case, it's bad. And also you can see affects the maternal care and the mom's health, with substantially affecting the baby. And unfortunately, breast milk highly accumulate PFAS. The mom's body actually detoxifies through breastfeeding. And where those PFAS went? It went to the baby, sadly. And so they're their smaller body. And so the concentration of PFAS in the babies are higher than adults when they expose the same amount of PFAS. yeah, it does affect our next generation more than adults.
Courtney, can you talk a little bit more? Your slides had a comparison of background to site. Can you talk a little bit more about how you thought about the differences and what the difference between those two are?
Yeah, sorry I had to blow through those.
Okay.
We actually designed that study for a few reasons. We wanted to be able to composite samples across farms to protect the confidentiality, to protect the identity of the farms. Um, and the chemists we were working with were sort of developing these methods for foods. So what we ended up doing, oh, and the other reason we did it was because we had heard that paper mill pulp may have been land applied on farms. And we thought we might be able to capture that using this approach. So we had a 3 mile radius around our site, where we like called that site. So if it was from a farm, we got the samples from a farmer's market. So we did our best to assure that they were coming from a farm within that site. And then our background locations, they had to be at least, sorry, maybe it was 5 miles from a known PFAS contaminated site. But I think that, what we're seeing with the signatures of PFAS in the squash and the background locations and in the site locations, so I don't know if you notice that sometimes it was higher in background, indicates that there's another source. So, land application of biosolids is really common. And as far as I know, there aren't records in Michigan of where that's been done. So we weren't able to sort of exclude farms that had that. So, or sites that hadn't been identified yet.
Right.
So background is like background, like we did our best. But you do see like a clear signature for some of the foods that we did have a sample, at least one sample that was really from our sort of hot zone.
Yeah. Well, and relatedly, there seems to be someone from North Carolina DEQ on the webinar who put a link in the Q&A of data recently released by the state on biosolids and comparing industrial, residential, and municipal. But I think, and Courtney, you started typing an answer to this in here too, but not all biosolids are the same. They don't all come from the same place. So do you know of anywhere that is attempting to figure out, like, if there's PFAS in biosolids, where that PFAS came from? Is there value in kind of the forensics in trying to figure that out?
Well, sort of the simple thing that Michigan did was and I'm sure there's reports on this. I'm sure I've seen them. But, they didn't know what was normal to some degree. And so they did a whole bunch of testing and then compared. And then it was pretty clear what was high. And the big signature that they were seeing was PFOS. And they were able to trace it back primarily to these plating facilities. So, yeah, I think states have been doing that. And it's not my area per se. But yeah, looking at those signatures can be a way to try to trace back. But it is complicated because then you have precursor transformation. So what you end up having in the environment can look different from what was at the source initially. Chris Higgins, if he was here, would have a really good answer for you. But Pingping, I saw a question that I wondered if you could answer better than I can, which was this: is PFAS chemistry in drinking water affected by water fluoridation? We get a lot of questions about fluoridation with PFAS, and they're
fluoridation is kind of a technique commonly applied in like water facilities, kind of to help to kind of prevent tooth decay. And that's an inorganic fluoride, so it's not PFAS. So the PFAS will not be impacted by the water fluoridation there.
Following up on what Lipeng was talking about a little while ago, do we have any scientific evidence yet that early childhood exposure to PFAS increases chances or susceptibility of adult disease?
Yes.
Period.
Yeah.
The answer is short, yes.
Yeah.
Mhm.
One thing I was interested in, I know it was on one of your slides where there was an arrow down and then we couldn't see the figure at the bottom, but it was related to PFAS and immune function, and you talked about cancer. And I had a colleague mention this once, and I found it really interesting. And I was wondering if you or Courtney or anyone else can speak to it. But we always hear people worried about cancer. It's the scariest health outcome in most cases, right? When we talk about exposure to something, we're worried about us or our loved ones developing cancer. But it's really hard to do a cancer cohort study. And so looking at immune function or other health outcomes that are likely to make you more susceptible to or can be initial symptoms of future cancer can be really helpful. So can you talk more about that and why understanding immune function can be important for understanding cancer?
Yes. I'm not experts in cancer, but what the principle is, our body using the immune system to constantly clean up our dead cells or mutated cells, which the original of cancer. But if our immune system is disrupted or immunity is down, then you lose ability to fight for your body to clean up the dead cells, clean up the mutated bad cells for original of cancer. So that's the principle of how immune system relevant to cancer development. I think B cells can be used as a biomarker for early caution, but I haven't seen any studies looking into it.
This is, go ahead.
Something like a genetic test. There are many genetic tests out there you can screen for cancer risk, but after your screening, you don't know what to do. There's chances you may have cancer. There are chances you may not develop into cancer because cancer development is complex. So what are you going to do? You just worry about getting cancer, or unless you have the concrete genetic mutation, the doctor will tell you you have a 90% of a chance to develop cancer. And you might do something, remove your organs, or in reproduction or in women's health, you can remove the breast, you can remove the ovary to prevent it. Otherwise, if we don't have a preventive treatments, we tend not, as a physician, we tend not to tell our patients, you have this risk, you have that risk.
Yeah. Well, and even on the opposite side of that, we see people living in impacted communities who do have cancers or other scary health conditions, and we can't prove that their PFAS exposure caused that. And that can be really unsatisfying as well. So, as much as we can learn from health studies and epi studies, there is always going to be that individual by individual uncertainty. And so, with that in mind, as we start to wrap up, here are some of the things that seem helpful that I heard, and then I'll ask you all for some closing thoughts. So, if you can find PFAS-free compost and you can use it in your gardening, it's a good way to reduce the potential PFAS in your fruits and veggies. We know that certain fruits and veggies tend to have lower levels of PFAS than others. So our water-rich ones are better. Is that right? Than starchy ones?
May have higher levels of
Oh, our water-rich, our juicy fruits and vegetables that we really want to eat in the summertime tend to have higher levels of PFAS. Courtney,
from a contaminated area. Like, I don't know
right.
that we can generalize across foods currently.
You saw that certain parts of the corn, like the corn kernels, didn't have measurable PFAS.
Yes, you're right. We can generalize about corn.
That's great. That's great news. We know that there are some strategies for removing PFAS from your body, like donating blood or the cholesterol medicine that Courtney mentioned. And there seems to be research continuing on new and upcoming strategies for removing it. One of the things that me and my colleagues who are organizing these webinars like to think about are like rules of thumb for communicating with people in the real world about reducing PFAS exposure. And so I was wondering, and I know this goes against your natural urges as scientists to make things black and white and oversimplifying, but as we wrap up today, what are some takeaways or rules of thumb you want to leave people with, thinking about PFAS in their foods and their health?
Can I go before I forget?
Yeah.
I didn't get to talk about like our chicken data very much, but interested in chicken eggs. So if you find that you have PFAS water contamination, if you address that and start giving your chickens clean water, it should reduce the levels in eggs. But if you have, it can be tricky if they're foraging because the PFAS are being taken up from the soils into the insects and they're also eating soil. So you can only kind of get it down so low. And there's some studies out of Maine that have looked at this as well. And then if you are giving your chickens fish feed, that will really increase their levels of PFAS. And so if you stop giving fish feed, the levels should come down quickly. The levels in chickens come down very quickly
That's
compared
great.
to humans. So
That's
that's
great
some good
news.
news, right? Like you don't have to kill your chickens. You just, don't eat the eggs for a while until the levels get down. So yeah, there's some things that you want to pay attention to if you have home chickens for PFAS and also for other contaminants like lead. I think maybe I don't have chickens, but people who have chickens, they're excited to have chickens because it's healthier in a lot of ways. But it's
And
also,
free.
the places that raise chickens, they're really able to control their environments and really, really minimize the amount of contamination. So we did look at eggs from grocery stores and we did not find any PFAS in those eggs
Great.
for what that's worth. And, we talked about the well water being an important thing to test because reducing your exposure through drinking water is one of the best ways to reduce your exposure since we get so much exposure from drinking water if there is PFAS contamination in it, particularly if they're moderate or elevated levels. So I'll let somebody else go. I could go on all day.
Yeah, I guess I want to emphasize like the most of the study we show today are from impact the community. So for general public, please don't feel panic about eating fruits and vegetables because we also did a market basket study before when we were trying to develop the method. We got produced samples from grocery store, at least not for ultra shorter chain. Like for the other PFAS, we quantified them and we did not detect like detectable levels of PFAS in them. So generally, I don't think like there's a risk of going to say a random grocery store and getting blueberries. I don't think that's gonna be like having increase your PFAS exposure. That's not the case. So it all goes back to like are those produced from an impacted area or not. If they are not, I think it's okay, or it's safe to eat more vegetables and fruits. And another thing is I remember there was a question regarding groundwater testing. I think the good and bad thing about groundwater is they don't tend to change easily. So if you have PFAS in the groundwater, they may stay there for a long time. But if you have clean groundwater, I guess it also means you don't have to like analyze them routinely, right? If some private well owners really want to get their water tested, like one test result may last for a while. You don't have to check it, say, every week or every month because that's groundwater. And because private wells are not protected under the Clean Water Act. So if you really want to protect you, like just to see if your water is clean, I guess it may worth it to get one analysis and see if you have detectable levels of PFAS to get to make sure you feel safe to drink or use the water.
Right. All right. Last but not least.
I have two messages to our community. First of all, advocate for stronger regulation. I didn't talk about that slides. In 2024, we had our first national regulation for PFAS levels in drinking water, but then it's gone in 2025. The water limitation in North Carolina drinking water is ridiculous. Some of the compounds, the PFBSS study is 2,000 ppt, which means no regulation. And the P spiral drinking water we study, we had a few town hall meetings to address that issue to our community, and they kind of have stronger regulation to the companies dumping PFAS into the drinking water. And now Pittsburgh's drinking water goes down, but then Durham drinking water goes up because they don't have a penalties. Based on the North Carolina regulation, there's no penalty because they're not exceeding. Even though the community has concerns, they don't have there's no consequences. That's the first message I have. The second message is supports research in this area because we need more and more to show, especially there are emerging new PFAS compounds, and we're still blank about their toxicities, especially the short chains. I thought PFBS is the short one, and Pingping actually show even more short chains and novel ones. We need more studies into it to provide evidence for toxicities.
Courtney?
Can I add 2 more points? Don't eat liver. If avoiding liver can reduce your exposure, especially if you've got more highly exposed, but probably in general as well. Like I personally don't eat pate because of the way that contaminants like PFAS and others will accumulate in the liver. We have a deer advisory here in Michigan. So there's been some testing of deer in highly impacted areas, and they've been found to be elevated and trigger advisory. Again, the liver was what was especially high in the deer. So that's a good way to reduce exposure. And then if you have a garden, I'm really into gardening, so I want to say this for all my gardening peeps. The fertilizer that you buy at the store, like wherever you get your fertilizer from, often it's made from sewage sludge. So paying attention to where your soils and amendments are coming from can also help you avoid PFAS in your garden. And of course, your irrigation water, kind of going back to the water testing thing. So, thanks.
Well, thank you all so much. This was so helpful. The organizers were just talking while you all were talking of maybe we should pull together a list of a lot of the resources and tips you just mentioned in these last 5 minutes, but in addition to some of the papers you referenced and other things. So in case anyone wasn't able to take notes throughout the whole thing, I think we'll work on pulling some of this together because I think it's really helpful, practical things that people crave when it comes to a daunting, overwhelming topic like PFAS. So thank you so much for taking time out of what I know are really busy schedules. We really appreciate it. Kiara, take us home.
Yeah, thank you all so much, truly. And I think that all of the questions asked and answered were really valuable. Hopefully that they were. In this last minute, as we wrap up, just want to remind everyone that our third and final webinar in this series will be taking place Tuesday, April 21st, so about a month from today, from 12 to 1:30 online again. So this is some information about what to expect from that webinar. That's our setting and communicating PFAS fish consumption advisories in other states. So we're calling this more sort of the state of communication webinar. And then a reminder to please fill out the survey if you are able to. Once the webinar closes, you will get a screen that looks like that on your right, and you can go ahead and take the survey that way, or it will come to your email. So thank you all again, and hope you have a great rest of your Tuesday. Take care, everyone.